Distinguished guests and all participants, once again, my name's Nyawira Muchane, a research scientist at National Museums of Kenya. I am here to give a talk on mushroom identification guide with the aim of introducing some skills important in mushroom classification. Welcome. Another important skill citizen scientists need to effectively contribute to macrofungi research and conservation is to have basic skills in macrofungi identification. And the first question we need to ask ourselves is, why macrofungi identification? Skills in macrofungi identification are very key because they enable us group these macrofungi species in different categories depending on their close relationships. This in return helps us communicate effectively to other scientists or to other citizen scientists about what we have found or about what we have discovered. Another reason why we need the basic skills in mushroom identification is to enable us participate effectively in mushroom research. When we know the mushroom species in our community or in our area, it is now possible to formulate hypotheses. It's also possible to formulate research questions regarding the groups of species in our community. And when we do this, we are able now to carry out an important research about their management or about their sustainable utilization. Another reason why we are supposed to have basic skills in mushroom identification is classification knowledge helps us know, and it also helps us appreciate mushroom diversity in our community. This makes mushroom collection and research exciting, especially when you see some species you have heard about, or when you see some species that you know about them. Another reason why it is important to have basic skills in mushroom identification is that it helps us discover new species in our area. We can only know, or we can only ascertain that the species we have documented has never been documented before, or it is new into science if we have some basic mushroom identification skills. Lastly, we need these basic identification skills to enable us discover economically important species in our community or in our areas. This will help us differentiate between edible and poisonous species. It will also help us uh, understand the species, the uses of the species community in our area. We can understand the mushroom species in our area that have medicinal properties that can be used for dye, that can be used to expand the mushroom industry. And when now we have this knowledge with ourselves, we are able now to contribute effectively to mushroom research and its conservation. Having known the importance of basic knowledge in mushroom identification, in the next lines, we are going to discuss the basic methods that are used in mushroom identification and that are used in mushroom classification. Basically, we have two methods used in mushroom identification and classification. The first method is the traditional method. This method utilizes the visible macroscopic traits as well as invisible traits, also known as microscopic traits. The method also utilizes the mushroom habitat or where the mushroom is growing, and it also utilizes the behavior of a mushroom, how the mushroom relates with its environment. Another method that is used in mushroom classification is the molecular method. This method utilizes the genetic traits to identify mushroom or macrofungi species. We all know that individual species are known to have a unique genetic background or it have a unique genetic information. This information is usually found in one gene region or it can also be found in a few gene regions. This genetic information, or what we are calling the barcode, when it is utilized, it helps us in identification of species, or it will help us in identification of macrofungi species. In this molecular method, DNA is extracted from corrected mushroom species, or the DNA is extracted from corrected mushroom community. After extraction of the DNA fragments carrying genetic information, or fragments carrying genetic background used in mushroom identification are amplified, or they are multiplied to produce many copies of the original DNA fragment. These DNA fragments, or these backgrounds, are thereafter sequenced, or they are read to know their chemical basis, and at the same time to know the sequence of these chemical bases. Now, to know the mushroom species corrected, so we have the comparison between the genetic information of a known species with the genetic information of known species. And now from this comparison, we are able to know the macrofungi species we are dealing with, or the macrofungi species we have corrected. Molecular methods of mushroom identification are gaining preferences by many scientists. The reason being the molecular mushroom identification method are very quick and also very accurate. The method can also be used to identify fungal community in an area without now correcting individual species. This means if I correct soil sample from a given ecosystem, it is possible to use molecular methods to know the fungal diversity in that area. However, this method, although they are very quick and accurate, they have several disadvantages. One of them, the method is very expensive, and here in Kenya, one sample can be identified at a cost of 5,000 to 10,000 Kenya shillings. Another disadvantage of this method is that only few mushroom species up to now have been sequenced. This makes it hard or difficult to find reference information to compare the sequenced DNA of unknown mushroom species. The second method used in macrofungi classification is translational method. This method utilizes both the macroscopic and the microscopic traits to identify the macrofungi species. In addition, the method utilizes the habitat information as well as the mushroom behavior. In the next slide, we are going to discuss the macroscopic traits used in macrofungi identification. We can say that the macroscopic traits are the traits that are visible, or they are traits that we can see with our naked eyes. These include the features of a cap. The cap is usually the upper part of the mushroom or the macrofungi. The second one is the characteristic of the stipe, and we say the stipe connects the cap with the mycelia, or it connects the cap with the substrate. And the other one we use is the nature of spore producing tissues. And we know in the spore producing tissues, we can have the gills, we can have the pores, we can have the ringes. So all these traits, when they are brought together, they help us classify and assign names to macrofungi species we have corrected. The first trait we describe in a mushroom specimen is the cap, or the upper part of the mushroom. In a cap, we reckon the color, the size, the shape, the texture, the moisture, the odor, and also the consistency. Mushroom caps usually come in different shapes and texture. The most common shapes we'll find in the field when collecting mushroom are the convex shaped mushroom, or the curved caps. We also come across conical shaped mushroom, or we also come across the bell shaped mushroom. We also come across the bonneted mushroom, or mushroom with a raised center. We also come across the plat shaped mushroom, and also we'll come across mushroom with a depressed caps or caps that are funnel shaped. Other shapes we'll find in the field will include fan-shaped cap, commonly found in polypus. We'll also find burrow-like shapes found in puffballs. We also found funnel-shaped fungi. We'll also find bird nest-shaped macrofungi, artista-shaped macrofungi, cap-shaped macrofungi, branched, and also coral-like shapes, among others. In the field, we'll also find caps which come in different textures. We'll find caps, caps which are smooth or caps with no ornamentation. We'll also find hairy caps. We'll also find caps with the raised scales. Others have flat scales, and others have patches of ornamentation. The correction of these shapes and texture are very key in mushroom identification. Another trait we use in macrofungi classification is spore-producing tissues, and we usually have two main types of tissues, the gills and the pores. The pores come in different color, they come in different shapes and sizes. For example, we have the angular pores, we have the round pores, and also we can have mushroom with the microscopic pores, the pore that we can not see with our naked eyes. We can also have mushroom with the tooth-like pores. In addition, we have spores that are produced in a ball-like structure, and this one is usually common with the puffballs. We also have spores that are produced in ridges-like structures, as we can see from this slide. Apart from the pores, we also have gills as a spore-producing tissues. Gills come in different colors, and it's usually very important to document the colors of the gills when you are in the field. In the gills, we are also interested in knowing how they are attached to the stipe. When we go to the field, we will find gills which are free, and these are the gills which are not attached to the stipe. We'll also come across gills that are straightly attached to the stipe. This form of attachment is usually called adnext. We'll also find gills that are running down the stipe. This form of attachment is usually called decurrent. We are also interested in knowing how the gills are arranged, and they come in three different ways. We can have grounded gills, we can have cross gills, and we can also have distance gills. Another trait we look in gills is the presence of the veins. Here we show a mushroom which have gills that are attached to the stem, and we can also see the veins that are running across the gills. Gills color, attachment, and arrangement are very important traits in separating amongst similar species. Here we see similar species, but they have different gills color. The first specimen is a Conosibe species with the brown gills, while the other is a Mycena species with a white gills. In this other photo, we have the Macroscopic species with the white gills, and in the other one, we have Chlorophorum species with green gills. The Macroscopic species is edible, while the Chlorophorum species is poisonous. This shows the importance of using gills to identify mushroom or to recognize mushroom in the field. Gills color may change with age, so when you are in the field, it's important to note whether the color you are reading from a mushroom it is from a young species or a mature species. For example, in this slide, we see Reratiomis gills changing from white gills in a young species, and the gills darkens and becomes dark brown as the species mature. It is thus very important to record gills color from both the young and mature species. Another macrofungi structure we describe to help us in mushroom identification is the stipe. The stipe connects the cap and the mycelia or it connects the cap with the sample trait where the mushroom is growing. The traits that we look for when describing the type in the field include the color and the texture. So when you're in the field, we check whether the type is brittle or firm. We check whether the type is smooth or hollow. We check whether the type is chalky or dusty. And we also check whether the type is curry or leathery. Another trait that we look at the type are the presence and absence of universal veil or the partial veil. Universal veil is usually a veil that covers the entire mushroom as it sprouts or as it grows. The veil usually ruptures as the mushroom grows. We will know that the mushroom was covered by a universal veil when we find its remnant on either the cap or the stipe. In the cap, the universal veil remnant is usually evidenced by its remnants called the warts. In the stipe, we have the socks-like structure called the vulva, and these two structures, when we see them in the field in a mushroom, it is evidence that the mushroom was covered with a universal veil. The partial veil, on the other hand, covers only the mushroom gills. The presence of the partial veil in a mushroom or a macrofungi is usually evidenced by the presence of annulus ring. The annulus ring come in different shapes. We have the pendant, the fraying, the sheathing, the double, the cobweb, and the ring zone. In this slide, we show photos of some common shapes of the annulus ring. Another trait we use in mushroom identification is the stipe shape, presence of the cuticle, and also the presence of mycelia like roots. The cuticle is usually the skin of the mushroom, and we can know whether the mushroom has a skin when we peel off. Mushroom types come in various shapes. We have the mushroom with an equal type. We have a mushroom that has a crab shape. We have a mushroom that has a bump, that is barbarous. We have a mushroom with a vulva. We may also have a mushroom that is deep-rooted. We also have the mushroom that have mycelia. All these are the common traits we need to describe when we are in the field to help us in identification of the mushrooms. Another trait that we need to check in the field is the presence of breeding or oozing of any liquid in a mushroom or when the mushroom is bruised. This trait is especially very important in identification of Ractaria species and some of the Agaricus species. Another trait we use in macrofungi identification is the color of spores or the color of spore print. Different macrofungi species produces spores of different colors. In this line, we can see how colors varies among spore print from different species. The spore print is usually very vital in separating different families. Apart from using the visible trait or the macroscopic trait, it is also very vital to use microscopic traits or the traits that we cannot see with our naked eyes in mushroom identification. One of the microscopic traits we use in mushroom identification is usually the size and the shape of the spores. At the same time, we usually like to check on the reaction of these spores with certain reagents or certain stains like MESA reagent. In this photo, we show different shapes, different ornamentation and colors of some mushroom species. Another microscopic trait we use to identify macrofungi is observation of spore-producing organs. In the Basidiomycota species, there are some species that produces infertile basidium. This infertile basidium is usually called the stidia. So the presence and absence of the stidia in any species is a very key structure that is used to separate species in the same genus. The third microscopic trait that we use in separating among species is the hyphae connection. There are some species in Basidiomycota phylum that form hyphae with the cramp connection. The observation of cramp connection, therefore, becomes a key trait in separating species from the same genus. Another trait used in discriminating against species is the habitat where the mushroom or the macrofungi is growing. We also use the behavior of this mushroom in relation to the environmental factors and also in relation with other species. Macrofungi species usually grow in different habitats. We have macrofungi species that are decomposers growing in dent tree stumps. Other grow in dent litter like leaves and twigs. Other grow in animal dung. Other decomposes the soil organic matter. Other species of the macrofungi are usually parasitic. They depend on the living organism for food. Others, they form symbiotic relationship with the plant species where both the fungi and the plant benefit. Other traits that we are interested when we are looking at the macrofungi is how these macrofungi are relating with the external environment. We need to understand the growth of these macrofungi in relation to the rainfall, in the relation to the amount of humidity, in relation to the temperature, and also the altitude. These traits are also very important in discriminating among species. Traditional method of macrofungi identification using morphological traits is the most common method used in macrofungi classification and identification. The method is preferred because it is easy to apply. The method can also be applied by a wide range of people. The method is also not very expensive. However, this method has several disadvantages. One of them is that the morphological traits can be very confusing and this can result in wrong naming. The method is also very tedious, therefore it is time consuming. Classification within kingdom fungi is not yet stable. There are several revisions undergoing. The current classification that was made in the year 2018, it divided the fungi to over 16 divisions or phyla. The macrofungi were classified into phylum, the phylum Ascomycota and the phylum Basidiomycota. The species from this phylum were separated mainly by the mode of spore production. Spore production from the species from Ascomycota phylum, they are usually produced in a necrosed structure called ascus. The phylum Ascomycota has about 64 species. These species are classified in 15 classes, 68 orders, 327 family, and over 6,000 genera. In this slide, we show some of the species that you may find in the field from this phylum. On the other hand, species in Basidiomycota phylum produces spores on a crab-like structure called basidium. The phylum Basidiomycota has over 30,000 species. These species are classified in 3 subphylum. In this subphylum, we have 16 classes, 52 orders, 177 families, and over 1,500 genera. Species from this phylum range from guild mushroom, jelly-like mushroom, polypus, puffball, bird nest, coral-like fungi, aster, crust like fungi, as well as stickenhorn species. From this talk, I have given some overview of the basic skills we may need to help us in macrofungi identification. We have also shown the importance of having these identification skills. We are currently working in producing a simple mushroom field guide to mushrooms of Kenya. This field guide will continue all the information we have learned from these two webinars. Those interested in receiving this guide when it's out, please share with us your email addresses and we'll contact you as soon as the guide is out. As I conclude, I wish to thank the iNaturalist team for supporting this webinar and for supporting this work. I also wish to thank the National Museums of Kenya, Nairobi University, and all Carnival staff for supporting this work. I also thank all of us for joining and listening. To all of us, thank you, and let's continue keeping the mushroom research ongoing.